Hello and welcome to the Galaxy video tutorial for copy number variation analysis using VarScan2. The purpose of this project is to create an easy-to-follow tutorial on identifying copy number variations using VarScan on Galaxy. So not everyone has the coding skills to analyze their data using code. So Galaxy is an amazing tool that allows anyone to access the tools programmers use to process their data without using any code at all. Cancer research is a large and interesting field of research that many students may want to enter after college. So being able to analyze cancer data is a critical skill for entering this field. The following tutorial will show you how to analyze copy number variations using a tumor and normal tissue sample taken from a real-world cancer patient. While this video tutorial starts from step 7 in the manual, steps 1 through 6 in the manual show you how to upload and pre-process your data. In this video, however, we will only be starting from map with BWA MEM, which is step 7 in the manual. The workflow for this can also be found in the manual. So the way that I will be showing you what I input these tools when creating my workflow is using this rerun button that could be found within your runs right here. And this is going to show you what I input into the tool when I ran it. So in search tools, in the top left corner is how you can find the tool that you want to use. So if I was trying to use map with BWA MEM, I could type it in and it will pop up. The reason we are using map BWA MEM and not just regular map BWA is because map BWA maps short reads and map BWA MEM maps medium and long reads, and that is the type of data that we have in this tutorial. So under will you select a reference genome from your history or use a built-in index, we will be choosing use a built-in genome index, and under using a reference genome, we will select human HG19. This is the genome that we will be using for all of our analyses, so anytime a tool asks you what reference genome to use, please select the same reference genome, human HG19. Under single or paired end reads, select paired. Under select first set of reads, you are going to select the trim-o-matic output for R1 or your forward reads for the normal data. Under select second set of reads, you're going to select the trim-o-matic output of your normal R2 paired data. You will have to run this map with BWA MEM twice, once for your normal tissue data and once for your tumor tissue data. What I'm showing you right now is for the normal tissue data. Under set read groups information, you will set read groups to SAM/BAM specification, and under read group identifier ID, type the normal tissue ID number 231335. Obviously, when you are running this for tumor reads, type the tumor ID number 231336. These ID numbers can be found in the manual. Under read group sample name SM, type normal, as this is the normal tissue data. When you're running this for tumor, type tumor. Run the tool by clicking Run Tool in the top right corner here, and this will create one output in your workflow. Obviously, you're running this twice, so you will have one for normal and one for tumor. The next tool we are going to use is RMDupe. RMDupe removes the duplicate reads that were produced during PCR. So we're going to run this twice, once for the normal data and once for the tumor data. When you look it up, it will say remove PCR duplicates. So under BAM file, choose the map with BWA MEM output for the normal data, and keep the default settings BAM is paired ended and treat as single end no. Run this tool once for your normal data and once for your tumor data. It will give you one output for each. The next tool we are going to use is BAM left align. Again, we are going to have to run this twice, once for our normal data and once for our tumor data. So BAM left align can left align our reads around indels. As stated on the GATK website, the same indel can be placed at multiple positions and still represent the same haplotype. So by left aligning these, we are fixing that issue and left aligning all of our indels. So under select file in BAM format, choose the RMDupe output for the normal sample. Using Under Using Reference Genome, choose the human Homo sapiens HG19 reference genome again, and leave the rest of the default options. Run this tool once for the normal sample and once for the tumor sample. This will give you one output each in your workflow. The next tool that we are going to use is SAM Tools CalMD. Okay. SAM Tools CalMD recalibrates read mapping qualities. The main reason for us using the tool at this point is to cap the mapping quality of reads before variant calling. So, under BAM file to recalculate, choose BAM left align for the normal data. Under Use a Reference Sequence, choose Use Built-in Genome and select Human Homo sapiens HG19 from the list. Under Additional Options, choose Advanced Options. And under Set the Coefficient Cap Mapping Quality of Poorly Mapped Reads, we're going to set that to 50 because this is the recommended setting for mapping reads aligned with BWA, which is how we mapped our reads with Map with BWA. You're going to run this tool once for your tumor data and once for your normal data. The next tool we are going to use is Filter BAM. And when SAM Tools CalMD was run, it will set undefined mapping qualities to 255. So these reads need to be removed from the data using Filter BAM. So under BAM Datasets to Filter, choose the SAM Tools CalMD output for the normal data. Under Filter Reading Map Quality Fred Scale, you are going to set that to less than or equal to 254. So this will remove all reads that have the undefined mapping quality of 255. There are other things that you can filter your data on, but we're just filtering on mapping quality today. You're going to run this twice, once for your tumor data and once for your normal data. Every time you run Filter BAM, it will output 2 files in your workflow. The next tool is SAM Tools MPileup. This is a really important tool to run when trying to use VarScan copy number. So this tool is going to generate a text pileup output for our BAM files. This file type is necessary for using VarScan copy number as that's the input into that tool. So under BAM files, choose the Filter BAM outputs for both the normal and tumor data. So we will be using multiple data sets and we will be picking both at the same time. Under Use Reference Sequence, choose Use a Built-in Genome and select the human Homo sapiens HG19 from the list. So that will create our MPileup data type. Next, we are going to run VarScan somatic on our BAM files. Running VarScan somatic is going to help us identify all the somatic variants in our data, including copy number variations, but not limited to copy number variations. This tool is going to output a VCF file. So under Use a Reference Sequence, choose the Use Built-in Genome and select the human Homo sapiens HG19 from the list. Under Aligned Reads from a Normal Sample, choose the Filter BAM output for your normal data. Under Aligned Reads for Tumor Sample, select the Filter BAM output for your tumor data. Under Estimated Purity for Tumor Content, change that to 0.5. Under Settings for Variant Calling, we are going to set it to Custom Settings. We are going to set the minimum base quality to 28 and the minimum mapping quality to 1. And you only need to run this tool once. If you're curious in seeing all the types of somatic variations that exist in this tumor data, you can click this eyeball icon and take a look at the VCF file that is produced. If you scroll down, you can see the different somatic variations that exist and how they're processed, but obviously this doesn't just include copy number variants. Next, we are going to run our VarScan copy number So this tool is going to output an interval file, and you need to input a pileup file. So this tool is used to identify which of the mutations in our dataset are copy number variations. So under how do you want to provide your pileup files, you are going to choose normal and tumor combined in one file because we combine these datasets in the mpileup tool. Under SAM Tools Pileup Dataset, which has combined normal and tumor samples, choose your SAM Tools mpileup output, leave the rest of the default settings, and run the tool. Next, we are going to use SNP SIFT intervals, and we can use this tool to match our VarScan somatic output with the VarScan copy number output. By matching these two files, we will create a VCF file that only contains copy number variations. So under Variant Input File in VCF format, choose the VarScan somatic output, and under Intervals Bed File, choose the VarScan copy number output. Run this tool without changing any other settings. Finally, we are going to run SNP EFF tool. So this tool annotates and predicts the effects of variants on genes and proteins. The output is easier to understand and contains some visualizations of what's going on in the data. So in the top, under genome source, choose locally installed SNP EFF database. And for genome, we're going to choose our Homo sapiens HG19. We are going to input this SNP interval tool output, which created a VCF file only containing copy number variations. When we run this tool, we will get two outputs, one of which being the HTML stats, which, if we take a look at, will give us visualizations of the different variations that are occurring in our data. There we go. So it will tell you the type of variations that are happening in our data, what effect by type and region. It'll create different histograms. You will be able to see the allele frequency and the allele count, codon changes, so how many times AAC was replaced by AAT, amino acid changes, what position the variants are. As you can see in all our data though, there are only variations being shown on 5, 12, and 17. That's because the data set that we are using, as said in the manual, has been downsampled to only those 3 chromosomes to make it easy to process in this tutorial. If you click that at the bottom, it'll break it down by what gene is being altered and how.